Hello, my name is Seth Temple. I am a postdoctoral research fellow at the University of Michigan right now. Today I'll be re-recording a conference talk that I just gave at the American Society Statistical Association for the section on statistical genomics and genetics. So without further ado, uh, I'll start this talk. It should be roughly 12 to 15 minutes when I gave it at the conference. Okay, so much of this is concerning my thesis research at the University of Washington Department of Statistics. Today, my topic is multiple testing corrections using identity by descent segments. So I'll start by just setting the scene of what these identity by descent segments are, which is a form of genetic data. Overall, I have and continue to develop methodology based on these IBD segments. So we can understand these in the context of a pedigree. On the left hand, I have the past, where we may have founder genomes, our great-great-great-great-grandparents. They have offspring, their children, and those children have a mosaic of these stretches of DNA that they inherited. And it's a mosaic because of this process called genetic recombination. Now, if we have many, many generations into the future, at each step of an offspring, we have genetic recombination that breaks down these stretches of DNA. Now, in the present day, when we look at the modern genomes who have many, many ancestors, they have these stretches of DNA that they might have still inherited identical by descent from these two founder genomes. If those segments, which I've depicted here in the black rectangles, are sufficiently long, then we can detect these with high accuracy. So for the remainder of this talk, I will assume that we have detected these IBD segments with perfect accuracy. So the goal here is that we want to determine genome-wide significance in a particular disease mapping study for complex haplotypes. Now, up in the upper right-hand side of this slide, we have this dichotomy of allele frequency by the genetic effect size. And we have multiple different types of studying such variants with different experimental designs. So what's very standard is this single marker genome-wide association study, which has a lot of power when we're thinking about common variants with moderate to high effect size. We also have rare variant studies where we don't see a lot of these variants in the population, but they could be highly impactful on a phenotype. So IBD-based disease mapping exists in the space of this red star. We may have multiple and possibly ungenotyped causal variants, and these will be at rare to low frequency, and we will aggregate the evidence of those variants. And all of those variants may have some moderate effect size. These can be particularly useful when we're thinking about understanding the mechanisms of disease as we trace back the inheritance from our ancestors. So throughout, I'll have figures that look like the figure here in the bottom of this slide. We'll be considering the 22 human autosomes on the horizontal axis, and on the vertical axis, we'll be considering some test statistic. And we would like to determine what this horizontal green line is, the genome-wide significance level, such that the loci exceeding that threshold will investigate further. So the outline of this talk is that I'll argue for a two-sample z-test as a particular way to detect case control effects in IBD mapping. I'll give an approach to control the family-wise error rate. So this is the probability that we make one or more false positive conclusions when we do multiple tests along the genome. And then I will apply our methodologies to the Alzheimer's disease sequencing project and discuss one known therapeutic target that we identified. So overall, our main test statistic we'll refer to as the IBD rate. I'll break down how we come to this test statistic. So we have the IBD segments that we're studying around some focal point, which is this black circle. In this example, I'm considering four haplotypes, little a, b, c, and d, and the shared DNA that they have. So the stretches of IBD are to the left and right of this focal position, which we'll denote as these random variables W. If these random variables, the IBD segment lengths, exceed some detection threshold, the gray line at the bottom of the slide, we're going to dichotomize such that we have these binary random variables denoted here as Y. The IBD rate is then the sample mean of the correlated variables. In this example, the sample mean is 4/6. A lot of our results will depend on a central limit theorem. So recapping, the standard central limit theorem that we have is if we have data that are identically distributed and independent with finite mean and variance, then a sample mean converges to this standard normal distribution. When we have such a result, it's quite useful for calculating p-values. So the IBD central limit theorem is work that I developed with Professor Elizabeth Thompson. It says that this sample mean, the IBD rate, is normally distributed under the following conditions. So we need a large sample size, little n, that come from a large population size, big N, and we're going to scale that by the detection threshold, W. The next condition is that the scaled population size should be much smaller than the sample size squared. So the probability of an IBD segment is of the order NW to the -1, and we have n squared, roughly, IBD segments to consider. So this condition is really just saying we have enough data that we actually observe some long IBD segments. The next is that the sample size needs to be much smaller than the scaled population size. This is what controls vanishing covariance terms, so that we don't have inflated upper tails. Recapping this result is that we have this quite useful theoretical result where we have some average in this statistical genetics study that is normally distributed under certain conditions. So we'll use this to propose the null model for a hypothesis test and calculate p-values. This CLT may be good, at least in humans. On the right-hand side of this slide, I've got the identity by descent rate histogram for a simulated scenario of human-like demography. Another useful thing when we have a theoretical result like this is that we may know when and how our null model could fail, or when our assumptions aren't working. So now I'll define our first hypothesis test in this research. It will be that we have this one sample z-test to detect excess IBD rates. The null model is that we have an average genome-wide IBD rate that is, in our case, going to be normally distributed. And then the alternative is that we have an excess identity by descent rate, and this could be due to the rapid adaptive evolution of a causal variant. We can make sense of this in the context of genealogies at a locus, otherwise known as the coalescent. So at this tree graph, at the bottom, we have our current samples in dark blue. They have common ancestors in the light blue, and the branches in this graph will be the time to these common ancestors. In the case of the alternative model, strong positive selection, those in red with the selected allele have more recent common ancestors. So there's been less generations under which genetic recombination can break down the shared haplotype segments. As a result, we'll observe excess IBD rates if selection is sufficiently strong. To determine a multiple testing threshold, we chose to model the correlations among IBD rates with an Ornstein-Uhlenbeck process. And we can use that both to come up with a fast simulation-based multiple testing threshold, or we also have a formula that's analytical to control the family-wise error rate. So two particular assumptions of the Ornstein-Uhlenbeck process. First is that at each locus, we need normal distribution of this IBD rate. So that's really the main motivation of our central limit theorems. And then next, we need exponential autocovariances between the different loci. And at least in the case of these shared haplotype segments, there's a lot of reason for this to be a good model. And we've done a lot of fitting to the data, and this does seem to hold. This is work with Professor Sharon Browning, my dissertation advisor. So now I'll use some of the methodology I've developed to analyze data from the Alzheimer's disease sequencing project. Recap, we've got our one sample z-test to detect excess IBD rates, and we're scanning the genome for recent positive selection. So on the horizontal axis, we have our genetic positions along the autosomes, and then here on the vertical axis, I've got our IBD rate, the sample mean. So I'm showing the IBD rates along these autosomes. In this case, we have multiple thresholds that we've derived that are by and large the same. And we can observe that there are some loci in European ancestry samples that are quite elevated. So two in particular are at the LCT gene, which allows people to digest lactose late in life, and then the multi-histocompatibility complex that is very important to the immune system. So both of these have long been known as selected loci in humans. Now we're curious about case control effects. We'll slightly modify our hypothesis test in this case. So we're again going to do a z-test, but instead this is a two-sample z-test looking for the differences in case-case IBD rates versus control-control IBD rates. The null is that these case-case and control-control pairings of the IBD rates are the same. And the alternative is that cases may have more IBD because they share a recent causal allele for the trait that we're studying. We can understand this in a similar way to our coalescent before. So we have the controls, and then we have the cases that before looked like a selected allele. In our case, it's selection in the form of sample ascertainment, that we've sought out individuals that have this binary trait. So here is another similar figure, but I have changed the vertical axis for the IBD rate difference when we look at cases and controls for Alzheimer's dementia. So here are our results for the European ancestry samples. The horizontal axis I've shown in green is our genome-wide significance level, and we use the same sort of multiple testing correction with the Ornstein-Uhlenbeck process. Now, a concern with our methodology is that we're picking up LCT and MHC again, and we don't believe that LCT is actually associated with AD. So strong positive selection, especially in Europeans, can confound our test. I'll conclude with discussion of this green star locus, which we think has a promising result. So this covers the NBAS gene, which is a known therapeutic target in the Alzheimer's field. It's been associated with a protective allele on the mitochondrial genome, and it's been proposed as a therapeutic target because of differential RNA expression. However, we have not found a single marker GWAS hit with AD for this gene. So our work, shown zoomed in on the right-hand side of the screen, has identified a genetic association with this rate difference of IBD. So in conclusion, we can use IBD rates to detect adaptive or disease alleles Um, the method is quite simple. We're just considering sample means and differences of sample means, and it's very easy to calculate such that we can scale to enormous whole genome sequence studies. The methods are automated in user-friendly pipelines, so you can run a single terminal command after setting up a configuration file, and your analyses should be synced up over time with cluster computing. And we analyzed with these automated pipelines 9 datasets from 5 different ancestry groups. You can use our methodology and software at github.com/sdtemple/isweep. So, in conclusion, I want to thank my funding sources. So, in particular, the Eric and Wendy Schmidt AI in Science postdoctoral fellowship, uh, the researchers and participants in the Alzheimer's disease sequencing project, my collaborators, Professor Sharon Browning and Elizabeth Thompson, and then Ellen Weisman and Elizabeth Blue with the ADSP. Okay, thank you for your time. Do I stop?